everyone. This is Dr. Anisha from the Department of Botany, St. Albert's College, Autonomous, Ernakulam. Today, we will look into a very revolutionizing technique in molecular biology called PCR. PCR stands for Polymerase Chain Reaction. The term might be very familiar because it is quite frequently used in biological context. Okay, so polymerase chain reaction, it is used to amplify or get a large number of copies of a DNA fragment under artificial conditions. We know DNA replicates within cells and we get copies of DNA so that when cells divide, they get equivalent amount of DNA from the parent cell. But when it comes to handling DNA out of the cell, for various purposes, we require a good amount of DNA or a good amount of a particular fragment of DNA. For this purpose, it is always advisable to have a technique by which we can get similar copies of DNA. PCR is a technique which came in 1983. It was put forth in 1983, and using this technique, we can get a very good number of copies of a DNA fragment. Even though we have a very small starting material, we can have a very large number of copies of that DNA fragment after the PCR reaction. So this technique, it was devised by a researcher called Dr. Carey Mullis in 1983. He came up with this technique and it became a revolution in molecular biology. These days, no biotechnology lab or molecular biology lab will be working without PCR. It has become an integral part of biological research. And this technique, as is evident from its applicability, it received Nobel Prize in 1993 for chemistry. Since it was a reaction, it received a Nobel Prize in chemistry in 1993. So, as I mentioned, we get a large number of copies of a DNA fragment using PCR. So, we'll just look into the components of PCR, what all goes into this reaction. So definitely, if I want to amplify or get copies of a particular DNA fragment, first thing that I need to have is the DNA template. So DNA template, it can be genomic DNA which has been extracted from some particular cell or tissue, or it can be even a fragment. It need not be complete genomic DNA. Even if a fragment that contains the part which we need to amplify is available, that will be sufficient. So first thing is DNA template. Then we need primers. So what are primers? Primers are nothing but short stretches of nucleotides or otherwise oligonucleotides, which contain something around 18 to 30 or even 40 nucleotides. And for one DNA fragment, we need 2 primers. So they will be binding to the ends of the DNA. Okay, they will be binding at the 3' end of the DNA. So one DNA molecule has got 2 strands. So one primer will be binding at the 3' end of one strand and the other primer shall be binding to the 3' end of the complementary strand. So primers are required. Primers will actually define the region that we will amplify. The next requirement is the DNTPs or deoxyribonucleotide triphosphates that go into the building up of DNA strands. Then definitely the master molecule is nothing but the enzyme which will carry out the reaction. DNA polymerase, and for doing PCR we make use of Taq polymerase, which is the DNA polymerase coming from the bacteria Thermus aquaticus. The speciality of this polymerase is it can withstand high temperatures till 95 degrees Celsius. It won't degrade at 95 degrees Celsius, so that it can be there intact for the whole reaction. Then magnesium ions are required for PCR reaction because it is a cofactor for this enzyme. So magnesium chloride is required. And finally, a buffer which will help in the activity of the enzyme is also required. So these are the requirements or components of a PCR reaction. Now coming to the procedure, there are three steps in PCR, starting with denaturation, annealing, and finally elongation. So as mentioned, it is a chain reaction. One reaction cycle, once it is over, it keeps on repeating. It keeps on repeating for 30 to 40 times, and this repetition gives us an amplified amount of DNA. So what happens in each of the steps? We'll look into. First is denaturation. Denaturation, the DNA template is subjected to, the reaction mixture is subjected to a very high temperature of 94 or 95 degrees Celsius, where the DNA strands will come off. They will denature. So the two strands will become separate. This is the denaturation part. The next step is annealing. In annealing, the primers, they anneal to the respective DNA strands. Now the strands are separate, so the primer can bind. The strands are in a single configuration, so the primer can come and bind. The primers, they will come and bind to the strands, and the annealing is carried out at a lower temperature, because if the temperature is high, the primers won't bind. The annealing temperature depends upon the primers that are being used, so it will be different for different PCR reactions, but generally something 40, 50 or 60 degrees Celsius, within this range we can get annealing temperature. That can be calculated depending upon the primers. So once the primers bind, the annealing part is over, and then the elongation happens. Elongation temperature is 72 degrees Celsius, whereby DNTPs are added by the Taq polymerase to the 3' end of the annealed primers. The primers, they are already in their position on the respective strands. In the annealing part, at 72 degrees Celsius, the Taq polymerase will add DNTPs to the 3' end of respective primers. So the strand will grow. So after each cycle, whatever was the beginning number of DNA molecules, it will get doubled. Okay, so in the initial phases, like if we start with one molecule, you will get 2 after one cycle, 4 after second cycle, then 8 after the third cycle, and it will go to exponential figures as the cycles proceed. And by the end of the PCR reaction, when something 30 to 40 cycles are over, a very high number of required DNA molecules have been formed in the system. So this is a very efficient technique. Whenever we want to work upon a particular DNA fragment, we can do a PCR reaction and amplify the number of its molecules. Coming to the applications, it is a widely applied technique, not just in research laboratories, but in other areas also. We can use it to amplify new genes. We can use it to study evolution using ancient DNA. Then it is also used in health sector for disease diagnosis, for early diagnosis of cancer, and also for detection of pathogens. It is also used in forensics for identifying criminals from crime sites. like that. So it is a very widely used technique owing to its versatility. So I hope PCR is clear to all of you. Thank you.